First of all, it's the last recording of the semester. Secondly, the last part of chapter 29 deals with inheritance. And inheritance is a fascinating topic that impacts us directly in our own lives. Now, we're going to go back and review a lot of facts about cells and organelles, about molecules in the form of genes, and try to explain what does that give us as a tool to understanding human inheritance. First of all, genes are carried on DNA. In fact, a length of DNA that makes one protein chain is the modern definition of the gene. The first time we dealt with this, and Mendel said, well, factors cause traits. And the Mendelian definition was a factor that produced one trait. So we talked about the gene for eye color. Now we know there's something much more basic, because sometimes that eye color protein is produced by the expression of different pieces of DNA. Now we call a gene that piece of DNA or that section on the chromosome that produces one protein chain. We kind of saw this when we talked about hemoglobin. Hemoglobin is a tetramer protein with 4 chains. And it has 2 that we call alpha chains because they come from the same gene, and 2 we call beta chains. They're the same but come from a different gene than the alpha chain. So in our body, in this human body, we have 46 chromosomes normally, except for our gametes. And those are 23 paired sets of chromosomes. Now, I can not only say I have 2 number 1 chromosomes, and by the way, I can say that because I've looked. In graduate school, I took a blood sample and grew some white blood cells and then collected cells that were dividing. And I was able to look at my own chromosomes, and I have 46. And I have an X and a Y. I saw that too. But 23 pairs, making up a total of 46. I can not only say I have 2 number 1 chromosomes, but I can say that one of those number 1 chromosomes arrived in the egg from my mother, and one of them arrived in the sperm from my father. That's the purpose of meiosis, to make that reduction division exact. That's why I say the product of meiosis is a cell with 23 chromosomes that is an exact half set of the normal chromosome complement of a human being. It's not just the number, it's the balance. And we're going to find later, with a few examples, anytime you play with that balance, any 45 number in a living human or any 47 number in a living human always involves some noticeable change in their physiology and in their health. This is something we've only been able to do for about the last 40 years, and that is to recognize the pairs of chromosomes and pair them up. You can see that across the top here, these are all similar enough in size that if they were all just stained dark on a light background, it would be difficult to pair this one with this chromosome. But with these banding patterns, for example, this length with a large block of red enhancement here, identifying that region definitely allows us to pair them into the 23 pairs. Now, when we present this in this way, the paired chromosomes with their banding patterns, we call it a karyotype. The word is right up here. This karyotype, as is usually done, is arranged from the largest chromosome and numbered. Number 1 is the largest, down through descending sizes. 18, 19, 20, 21, 22. Notice the 21. See how small it is? 21 is just a little bit bigger than the Y chromosome. But this is the one that causes Down's syndrome, when there are 3 present. Here's 22, and then by convention, we put the sex chromosomes last because there is an X and a Y. This is a male karyotype. You can see that from the chromosomes. If there are 2 X's here, it would be a female. Now, what can we see? We can see the chromosome pairs. If there's a change to the chromosome, for example, if there's a deletion, one section of arm 8 is missing, we would be able to tell. If there's a duplication, this arm is longer because instead of 2 red bulbs, there are 3. That duplication of genetic material, we would be able to see. But what can we not see? We cannot see the genotypes that are actually carried. So if we're talking about something like red-green color blindness, with big A representing the normal protein for being able to see the difference between red and green, and little a means you can't. I can't look at the location on the chromosome and see big A, little a, or big A, big A. That we cannot do. Patterns of inheritance depend on the chromosomes and how they move. We're looking here at a pair of chromosomes. This is what we call homolog. This is, if you want to think about this, is the number 1. This is the other number 1. And what we're seeing is a situation indicated by the colored dots that there's a location for that gene and that they're carrying different alleles, big A, little a. Simple inheritance is a pattern of inheritance where a single pair of alleles, one gene with 2 copies in the cell, is determining the trait. And that's true for about 80% of our genes and proteins. One gene makes the entire trait. However, many other traits, and some traits that we're interested in, some traits that we commonly think about, are determined by polygenic inheritance. This is where, now, to draw polygenic inheritance, we'd have to start with this, and over here we might draw the number 8 chromosome with 2 different alleles, and even not stop there. True polygenic inheritance or quantitative inheritance, there might be not 2, but 4, 6, 8, 10, 12, or more alleles that contribute to the actual trait that the person shows. So hair color, blonde and red hair are recessive traits, and therefore a single genotype produces them. But other hair colors, all of those shades from black to brown to auburn, those are polygenic traits affected by several genes. Skin color, that basis of that first cut for the basis of racism. Eye color, which interestingly enough varies. We recognize that people have different colored eyes, but for some reason, we're pretty tolerant of that. I mean, I'm not sure that having brown eyes or blue eyes or green eyes or hazel eyes or nothing eyes or all the other different kind of descriptions we have means anything particular about the person. And in fact, that's the way we typically look at eye color. Height is something that's important because height does represent a physical trait that is important to people. In fact, we have a whole array of fashion and clothing items that are popular because they adjust the height, typically increasing it. So polygenic inheritance is the other 20%. Over here we have a special example, sex chromosomes. We recognize the male, and by the way, for females, X-linked genes behave the same rules as we have over here, simple or polygenic, based on the X chromosomes, X genes that are present. But over here, since the male carries X and a Y, there will be a different pattern of inheritance since the X only carries many alleles that are not on the Y. So one copy of the allele, in this case red, whatever that red allele is, if that's red-green color vision, and this male will be able to see the difference between red and green. And red-green color blindness, some forms of hemophilia are carried on the X chromosome, and a particular kind of muscular dystrophy, Duchenne's muscular dystrophy, is also carried on the X chromosome. Now, because of this difference, comparing two homologs, what are called the autosomes have homologs. Over here, this is a heterologous pair, so this genotype is called hemizygous. This one would be called homozygous, or in this case that's illustrated, heterozygous. Those are the terms you may have used before. So it's called hemizygous. Because of this single fact that males carry only one X chromosome, that means that these traits will appear more often in males and actually be seen as male traits. So colorblind, there was a trait called male pattern baldness that we always thought was on the X. Then we find it's subject to modifiers, so we no longer list it as a primary male trait, but you should recognize that men go bald more often than females. Once we get the chromosomes defined, which is what the karyotype allows us to do, the Human Genome Project took that karyotype and placed specific traits, specific genes, means the production of specific proteins, onto the chromosomes, produced a map. And now we have to think about the combination of alleles for a particular gene. This shows two different alleles on the autosomes. So what does that mean? If it's simple dominance, that means the presence of one allele completely covers up the presence of the other one. This typically results because the red shown here will produce a good functional protein that does its job, whereas a mutation has changed the yellow allele, so it produces a protein that no longer works. Now, it's easy to see this if you think about pigments. If you have a pigment that is produced by a big A, and you have a mutation, so it changes the pigment, often that protein has no color. So little a would produce just an absence of the pigment. Examples of dominant traits and their recessive counterparts. So normal skin pigmentation is based on dominant alleles. Albino is a recessive trait at one gene. Freckles, absence of freckles, nearsightedness and farsightedness are both dominant. Normal vision is recessive. Down here we see other vision effects. Astigmatism is dominant to normal vision. There are a number of different mutations that are dominant to normal vision as we define it. Free lobes are dominant to attached. Tongue rolling is dominant to inability to roll tongue. This is an interesting kind of multi-muscle and nerve system, but it responds to a strict dominance inheritance. Sickle cell anemia is a recessive condition. It is recessive to the normal beta chain phenotype. The Rh factor is dominant to a recessive condition, Rh negative. Type A and B blood are dominant to type O blood. Huntington's disease is a dominant genetic condition. We'll talk about it in a minute. But cystic fibrosis, Tay-Sachs, and fetal ketonuria are all recessive conditions. So here we have a situation where if you're going around with normal type A blood, we don't know if your genotype is big A, or if it's big AO. You could be either one since A is dominant to O. Sometimes we can figure that out from the pedigree, the blood types of your parents and or the blood types of your children. Another type of expression is codominance, where both alleles, the presence of both alleles, red and yellow, show up in the trait. So type AB blood is a good example. They are codominant. That blood type results from the mixture. Albumins, the blood proteins primarily responsible for osmotic balance. The structure of transferins. And you notice right here, this is a short list, but we've gone from rather organism level traits like a blood type down to the specific structure of proteins, and more we're going to be learning about the genetic expression at the protein level. Incomplete dominance is something where the two genes, the two alleles, red and yellow, produce traits that are between the dominant or the recessive. Hemoglobin A production is sometimes shows this. Hemoglobin S production shows this. Now, hemoglobin S is the allele for sickle cell anemia. And if you remember, if you have the two copies of the beta gene, if they are SS, and you don't have any good beta chains and that person dies. If you have, however, a beta S, you get a level of disability. These people need to stay out of low oxygen conditions like unpressurized aircraft or the tops of mountains. But they still are viable, and that's an intermediate condition between the SS genotype and the BB genotype. When we look at the typical way we analyze a Mendelian inheritance, we typically set up a controlled cross. Now, this is how we study genes using experimental organisms. Those experimental organisms might be what were selected because they're easy to obtain, easy to grow, and easy to cross and produce offspring. So if you've ever wondered, why do we have so much information about fruit flies? Well, there's a number of reasons for that. Number one, we kind of don't find them in nature, but they're easy to collect. When we started studying them, we had these rest stops with open trash cans. People would put their garbage and their lunches and their trash in there, and in the summer, fruit flies would collect. So if you wanted to collect fruit flies, all you had to do is take your insect net and go over and literally kick that can, wait 2 seconds and wave across it, and you had a sample of fruit flies from that region. Once you get them into the lab, you can take these little half-pint glass milk bottles or cream bottles with a paper cap, put in a defined medium that has all the nutrients and water, and put 2 fruit flies in there. They'll mate and deposit eggs in 20 days. That first generation will be flying around in that bottle. So it's very easy to do crosses with fruit flies. We now do the same thing with microorganisms. And the advantage of microorganisms is you can not only do it in a matter of days, fewer than 21 days, you can study both prokaryotes and eukaryotes. And another thing you can do is you can generate literally thousands of offspring. So you do the gene transfer, and then you let the culture grow up, and you can see if there's a distribution of different traits. Now, in humans, we have to rely on pedigrees, but often they tell us something. Here's an example of a Punnett square analysis. Now, up here, we have 2 eggs labeled little a, little a. This is a mother carrying a recessive trait. So we see that she's expressing the recessive trait. Little a, that's all she can contribute. And here we have the sperm from the father, and the father carries the dominant trait. Now, when you have this cross, a dominant parent trait and a recessive parent trait, you always have a dominant allele in the sperm, so you can see just by combining, making all the possible combinations. Every genotype is big A, little a, and because this is a dominant trait, normal skin pigmentation is dominant for what we called albinism. Every offspring from this trait, from the albino mother, will show normal skin pigmentation. So it appears in our normal examination of families that albino simply disappears in this mating. If you do a different trait, let's say you have the same mother, see the little a, little a? Now, the mother's genotype is little a, little a. When she does meiosis, the two chromosomes separate, so they go into different eggs. But every egg carries a little a. That's what's shown up here. The reason we do it in double copy is that you can have a different genotype. You can be big A, little a, as you see over here with the father. Now here's a father who is heterozygous for skin pigmentation. The big A sperm will be 50% of the sperm population, and the little a sperm will be 50%. Now, looking at the father, you have no idea that that father carries this allele. The best clue would be if there's a history, a pedigree where you're looking at a brother or sister, you're looking at an aunt or uncle or parent or a grandparent in that generation, great uncles, great aunts, and you see an individual with albinism. Now, if it's in the direct line, meaning if the albinism appears in a grandparent, then it could descend to that individual. That individual could be heterozygous. But this trait, this cross, would tell you for sure. This is called a test cross because you're taking a heterozygous individual, the parent, that is the father on the left, and crossing it to an albino mother. Now, in this case, every fertilization that results from the sperm on the top is big A, little a, and these children are all normally pigmented. However, notice they are also all carriers of the albino allele, which means they can pass it on to their offspring. But down here below, because the father is a heterozygote, sperm can have the little a allele. That means that 50% of the fertilizations will be recessive and show albinism. Now, I've done genetic counseling in the past, and people interpret this in a kind of an odd way. So, in a situation where we had a parent, mother or father, who had a recessive trait, and a parent who had no recessive trait, had the dominant trait, there will always be the question of whether this parent is heterozygous or homozygous. If this parent is homozygous, then all of these individuals will show the normal trait. And that's good enough for most parents. But all of them, as we saw in the previous slide, all of them would also be carriers. Now, I don't know if I have the third cross, because this is clear that this makes it clear that this male is heterozygous. But what if the male is heterozygous, big A, little a, and the female is heterozygous, big A, little a? Now only one box right down here would be little a, little a. And this happens more often than you would think with rare traits. Let's say a trait is present in 1 in 100 individuals in the population. In a population our size, that means that it's going to occur that a man and a woman marry without realizing they carry the recessive allele. They don't have any history, but their first child shows the recessive trait. Now, if I actually diagrammed that out, you would notice that the chance of showing the trait is 1/4, one box out of 4. And the way people confuse those boxes is they come in to confirm. I've had people say, well, the chance of having the recessive trait is a quarter, and we had the recessive trait, so now we can have 3 more children who will be normal. And that's absolutely a misinterpretation of the chance of N. For a second pregnancy, the chance of having the recessive trait appear again is still 1/4. And the chance of a normal trait is 3/4. Going on, we see kind of the purpose of sex is to combine the chromosomes of different individuals so that in the cell, when we're looking at mitosis and meiosis, they can actually break and rejoin. What we see in the dividing cell is that these chromosomes lined up in pair. That's what we saw in meiosis, the homologs pair after they've been replicated. We can actually see in the light microscope, see these crossovers. And what happens is that at this point of crossover, but more important than that, it's not just at that point where the pointer is. There's an exact register. So if you have gene 394041, 394041 along the arm, if you break this strand between 39 and 40, then you break this strand between 39 and 40. So in most cases, there's an exact exchange of the arms from that point down. But look at what you have after this, why we call it recombination. This outer chromosome, where the pointer is, the dark purple one, is the same chromosome that came over from the mother or the father. This blue one is the same chromosome that came on from the other parent. But these two in the middle have been recombined. They are brand new. So this chromosome has been changed from the parent 1, which it is down here, and then the lower arm, about 1/4 of the chromosome, is parent 2. And the opposite is produced over here. Now, when you think about this crossing over that you see, we might break it here in this cell. The next cell, we may cross over up here. The next cell down here. In some cells that you look at, you will see not one crossover, but multiple crossovers, with this arm crossing with this arm, and these two arms crossing over maybe down at this position. So when we study meiosis and we look at a lot of those cells, we'll count how many crossovers occur. So the crossover frequency on that chromosome might be 2.7, meaning we're producing down here not just a bicolored chromosome, but one that may have 3 or 4 alternating bands. Now, the number isn't that important. The realization is, what am I doing? We're not even keeping the chromosomes from the parents intact. We're mixing and matching those genes and the alleles they carry into brand new combinations. And this and the separate chromosomes segregating away from the plate and to different ends of the cell produce an endless variety of genes in the gametes. And that's the point. Sex produces variety. Since I'm obligated to use sex if I want to reproduce, I not only am going to produce a new mixture, it is impossible for me to reproduce the exact genotype that I have. That would be called clonal reproduction. And that's one of the great debate points about how we apply the technologies that we've developed for cells and for the engineering of genes. So let's take a look at an X-linked trait. We mentioned that earlier and said that it was unique for males and females, in males and females, for genes carried on the X chromosome. Let's take a look at an example of that. Here we have a colorblind instance with the big C allele shown here and here. being the normal allele carried on the X chromosome. They have color-coded the X's so that you know whether it comes from the mother or the father. In this case, we have two individuals who have normal color vision. You can see that the female carries one big C but one little c. She is what's called a carrier, normal vision, but carrying a recessive allele on the second X. Over here we have a man who has normal vision. He has only one X, and it carries a big C. The Y has no gene that affects color vision. So let's just do our square. Let's bring the gametes down from the top and across from the left. And you'll notice because the sperm always has a big C, it makes no difference to your color vision which allele you receive from the mother. This is a normal female, but also a non-carrier. Her offspring will never receive the color-blind allele from her. But over here we have a normal color vision female, her sister, who has a little c, so she's what we call a carrier. Her vision is normal, but she can contribute a little c to the grandchildren. Down here we have a normal male, and if you are a male and the sperm carries the Y chromosome, then the color vision trait is going to be determined by what's in the egg. So here's a normal male. Now a male can't be a carrier. If the person has normal vision, then you know the one copy of the X that's present in the male must have the big C. So no color implications for this man's children. But over here, because the egg carried a little c, the X chromosome must carry the little c, and the Y chromosome makes it a man, and so this is a colorblind male. Now, if you look over here on the left side, this left column, there aren't any colorblind implications for this female genotype or this male genotype. But over here, you can see this daughter, half of her eggs will carry the little c allele. And so half of her offspring will be colorblind if they are males. Half of the males will be colorblind, and half of the, let's say it this way, half of her offspring will be males who are colorblind, and half will be normal males because she carries the same genotype as her mother. Down here for this colorblind male, if you're a colorblind male, all of your daughters become carriers because the thing that made them the daughter is getting this X chromosome. This is why you often see a trait that's X-linked, like colorblindness, skip a generation. So grandfather was colorblind. A daughter with normal vision will have a son who's colorblind. And that's a pattern you frequently see for recessive X-linked traits. Final thing we want to mention, which is the threshold event for your generation, is the Human Genome Project finally getting our knowledge of chromosomes and maps and genes and codes and proteins all together to the point where we have now determined the genetic code and the genetic location of every gene that's needed to make a male, to make a female. We know how they're arranged on the chromosome. We transcribed the entire human genome, and we have mapped the location of literally thousands of genes, discovered a number of surprises. It leads to this concept of a genome. What is the set that represents the full complement of genetic material needed to make a person? Now here we're thinking more on the biochemical level. That genome is a set of genes. It can be arranged in different chromosome numbers or different chromosome arrangements, but you got to have all the genes to make a human. Karyotyping is the determination of the individual's chromosome complement, looking for chromosomal changes, but also thinking about the mapping and what chromosomes are carried together on the same chromosome. That leads to this figure. It's hard in science to actually accept or understand what an astounding figure this is. So that within a matter of decades, we've not just determined the banding patterns, for instance, the number 1 chromosome you can see here, all the way around through the 2, 3, up to the 21, 22, and here's an X and Y. This is presented as a human map, chromosome map, because, and this would be for a male karyotype, see all the banding patterns have been drawn on the chromosomes, and the specific location of genetic traits with known mutations have been mapped. So this is something that literally was impossible 15 years ago because we didn't know where they were. We were working on linkage data, but just this very limited map gives us some pretty interesting information. So depending on what you've heard of, have you heard of muscular dystrophy? That's located right here in the top arm, the A arm of chromosome number 1. That's where that gene is located, and the mutation that causes muscular dystrophy, meaning the allele, typically a recessive allele, that is located. Prostate cancer, the next thing, prostate muscular dystrophy. That I followed the wrong line. That's over on the X chromosome. So it's going to have an effect that's sex-linked. Prostate cancer is located at this location on the number 1 chromosome. This is where we would activate a gene, and mutation would begin a tumor in the prostate. Looking down this line, you can see some familiar names like Huntington's disease, cystic fibrosis, malignant melanoma, ovarian cancer, sickle cell anemia, which we've discussed at length. Chapter 19 is where we discuss it, is on the very end of chromosome number 11. This is what produces the beta chain in hemoglobin, and the mutation that causes the cells to sickle is located on that chromosome. We are now beginning to study these diseases in a brand new way. But what we can do is we can actually chop this up and put the gene that is mutant for something like breast cancer, your number 28, we can put that in a test tube. We can use that piece of DNA to make the protein and study how it's different. That will lead us to strategies on how to deal with people who experience this condition. So, hallelujah. Chapter 29 is done, and our last recording is in the bank.